Okay, everybody, this is a video about nucleic acids. It's one of the ones we couldn't go over in class because we ran out of time. Luckily, it's quite a bit of review from general biology. So here we go. There's two major types of nucleic acids, one being DNA and one being RNA. And DNA is probably the one that most people are familiar with and remember, and you know that this contains all of our genetic information. Okay, and we know that this is where our hereditary information is stored. This is the molecule that changes when mutations happen. And this is the code for all proteins made in the body. So that's what genetic information is. It's just a code of which proteins are made and when. And we know that those proteins can go on to make many other molecules. And namely, these are going to code for enzymes that go off and do other things and make other things. And then this right here, this is ribonucleic acid. So this is simply a copy of DNA. So a single-stranded copy. And remember, this one doesn't have T. So instead of thymine, it has uracil in RNA. So this is a copy of the DNA. Okay, so you'll remember there was messenger RNA. Okay, and messenger RNA is very temporary. So RNA doesn't last very long, whereas your genetic information, that lasts your whole life. It's temporary. Okay, there we go. So really one main function for both of these. All right, now let's take a look at the molecule. And so this is DNA, right? And this is just one monomer. So a monomer of a nucleic acid is a nucleotide. Okay, so that's the monomer. And so let's take a look at what these two look like. They look pretty similar. We've got a phosphate group, and actually let's label these. So this is phosphate. Okay, and this right here, and it's on both, this is a sugar. Okay, specifically this is ribose. Okay, over here this sugar is deoxyribose. So DNA has a different sugar on it. And this is what we call a nitrogen base. Okay, now these look exactly the same. So this must be either A, G, or C. Okay, so and remember, in RNA there would be U. Okay. All right, so these are the three parts that compose a nucleotide. The difference between DNA and RNA nucleotides, one being that there's a possibility of U for RNA, and we do T's for DNA. And notice this right here. That oxygen is there. It's present on the RNA nucleotide. On the DNA nucleotide, it's missing, so it's been deoxygenated. It's missing. It's not there. So that's why it's called deoxyribonucleic acid, and it's deoxyribose because it's missing that. Okay, whereas this is just plain ribose. So make sure you're able to identify the difference there. All right, so this is a picture of all four nucleotides, so the A, T, G, and C. And so you might remember that A and T always pair together, and G and C always pair together here. And so just be able to recognize sometimes it's two rings with nitrogen in it, sometimes it's just one, but you're going to want to locate that phosphate and that sugar, and as long as you see some ring with nitrogen, that's a dead giveaway that this is a nucleotide. Okay, so this is a cytosine nucleotide, thymine nucleotide, and so forth. So let's take a look when it's all together. So take a second, pause the video, and count how many nucleotides you see. You should have found 8. 1, 2, 3, 4, 5, 6, 7, 8. Okay, so this is the thymine nucleotide, right? Oops, I should have included that phosphate. Okay, so this is one nucleotide. Okay, specifically this is the thymine nucleotide. You will not have to know the differences between adenine and thymine, for example. You just have to know they go together. You know, it's a type of nucleic acid, or sorry, it's a type of nucleotide. Okay, so as far as DNA goes, we're very familiar that it has that ladder shape. What you need to know about that is that the sides of the ladder are made of alternating sugar and phosphate, sugar, phosphate, sugar, phosphate. And you should know that connected to the sugars are the steps of the ladder. So the nitrogen bases are found in the middle. They are hydrogen bonded here. Okay, and there's a specific reason why this is not a covalent bond. You might think, wow, DNA, that really should be covalently bonded. We don't want it to fall apart. But in reality, we do want it to come apart here because we need to copy it. And it makes it really easy to unzip during DNA replication. So what we want, though, is that the DNA sequence is stable. And so you notice the sequence of A's, T's, G's, and C's, that's all held together by covalent bonds. So the sequence is pretty hard to break up, but the two sides of DNA come apart and reattach quite easily.